Hey guys, Miss Cote here, and this is the first video in the cell unit. You guys have been learning about cells since middle school, so this chapter should be more of a review. And while I'm not spending time in class on this, you will be tested on this material. I expect that you watch these videos, especially if it's been a while since you've taken biology. In this episode, we'll review some basic knowledge about cells, the similarities and differences between prokaryotes and eukaryotes, and the importance of cell size. So let's get started. You probably learned at some point about the cell theory, which goes like this. 1. All living organisms are made up of one or more cells. 2. Cells are the basic unit of structure and function. It's the smallest unit of matter that's alive. 3. All cells come from pre-existing cells. And 4. While cells can be different from each other depending on what their job is within the organism, they all share common features. What we know about cells has come from scientists whose job it is to study the inner workings of cells and how they do what they do. Obviously, cell biologists can study cells through microscopy. However, the section on microscopes is not in the scope of the class anymore, so you don't need to know the differences between the types of microscopes. I do want to talk to you about one technique that's frequently used, and that is cell fractionation. I do see questions where, to find the answer, you need to be familiar with how cell fractionation works, so I just want to spend a little time talking about it. For cell biologists, cell fractionation is the way you can isolate a specific type of organelle. They use a machine called a centrifuge to accomplish this. Some of you may have already used a centrifuge in a Project Lead the Way class. We'll be using them later on in our course. A centrifuge's job is to spin at various speeds, and the spinning will apply centrifugal force to substances and separate the components by density or size. So in the case of cells, you can take a sample, throw it in a blender to break up the cells, and the result is a mixture referred to as a homogenate. The homogenate is what you will put into the centrifuge to spin it down into its parts. In this example, the first spin will cause the largest components of the cells to settle at the bottom and form a pellet. So if you want to study the nucleus of cells, these will be found in the first pellet. All of the other components remain in the liquid above it. This is called the supernatant. So if you work with another organelle other than the nucleus, you can pour off the supernatant into a new test tube and spin it at a higher speed for a little longer time. The next pellet that forms would contain the mitochondria and chloroplasts. If this is what you study, then you would use that pellet. If not, pour off the supernatant and keep spinning. The third component that would separate out would be the membrane components, such as endoplasmic reticulum, Golgi, lysosomes, etc. And the final component would contain the smallest organelles, which would be the ribosomes. Hopefully you can see that this is a great technique to separate organelles from each other to concentrate on one particular type of organelle. The next section compares and contrasts prokaryotic and eukaryotic cells, which should basically be a review for you. College Board still likes to ask questions about this to make sure you know the differences. Remember that on the most basic level, cells can be classified into being prokaryotic, which are essentially bacterial cells, or eukaryotic, which are all other cells, including ours. When it comes down to it, no matter what type of cell we're talking about, all cells have the same basic features. All cells are made up of a phospholipid bilayer, the cell membrane or plasma membrane. It could be referred to as either one. Inside that plasma membrane is a substance called the cytosol. Remember that the substance itself is cytosol. When you're describing the substance and the organelles that are floating around in the substance, then we call it cytoplasm. All cells have genetic material, DNA and RNA. The chromosome structure may be different between prokaryotic and eukaryotic cells, but they're still the structures that carry the genes. And all cells have ribosomes. If you remember, ribosomes are not bound by a membrane, so they're found in both types of cells. Even bacteria need to make proteins in order to do what they need to do. And again, as far as the differences between the two types of cells, you should have learned this in honors biology. Let's look at this bacterial cell as we go through them. Prokaryotic cells are the smaller, simpler cells. They were the first type of cell to exist. One major difference between prokaryotic and eukaryotic cells is that prokaryotic cells are characterized by having no nucleus. Instead, their DNA, which is mostly contained in one giant circular chromosome, is found in a region called the nucleoid region. Hopefully you remember that eukaryotic cells have a nucleus. It's probably the most prominent organelle of the cell, and that's where eukaryotic cells store their DNA. The second major difference between the two types of cells is that prokaryotic cells do not contain any membrane-bound organelles. If we look at our example of a bacterial cell, there's really not much inside of it. It basically has the chromosome and these little dots, which represent the ribosomes. Our cells, on the other hand, have all of those membrane-bound organelles that you've studied over the years. Endoplasmic reticulum, Golgi, mitochondria, lysosomes, and so on. There are other differences, but those are the main two. As we move through the course, we'll discuss any other differences that we come across. While eukaryotic cells are the bigger of the two types of cells, they have limits as to how large they can get. The main limiting factor is the plasma membrane. Shown here, the plasma membrane is a phospholipid bilayer that is studded with proteins. This structure is a selectively permeable barrier that allows oxygen and nutrients into the cell while releasing carbon dioxide and other waste products out of the cell. We'll look at the plasma membrane in more detail very soon when we get to cell transport in Chapter 7. The metabolic requirements of bringing nutrients in and wastes out is what keeps a cell from getting too large, and it has something to do with something called the surface area to volume ratio. The surface area represents the outer boundary of the cell, or the cell membrane, while the volume represents the insides of the cell. Only so many molecules can cross the plasma membrane in a given amount of time. This makes a small-sized cell very efficient at moving materials in and out of the cell. However, as the cell gets bigger, the inside volume grows faster than the boundary around it. In other words, as the cell gets bigger, the surface area to volume ratio decreases, and the plasma membrane cannot meet the demands of the cell in regards to supplying it with the right amount of materials. Let's take a look at how this happens. The first cell on the left has a cube shape where all sides have a measurement of 1. Let's say unit because it doesn't give us a specific measurement. To find the surface area of a cube, that would be the height times width of the side, so that's 1 times 1, which is 1, times the number of sides, which is 6. So 1 times 6 equals 6. To find the volume, that would be height times width times length times the number of boxes. So in this case, it would be 1 times 1 times 1, which is 1, times 1 box, which is also equal to 1. So if the surface area is 6 and the volume is 1, the ratio is 6 to 1, or 6. Just a side note, you may be asked by College Board to calculate surface area to volume ratios of cells that could have the shape of a cube, rectangle, or sphere. The formulas to do these calculations are on your formula sheet, so you do not need to memorize this. You just need to know which one to use and how to use it. Looking at the next cell that's larger and has a measurement of 5 units, the surface area would be 5 units times 5 units times 6 sides, which is equal to 150. The volume would be 5 times 5 times 5 times 1 box, which is equal to 125. Dividing the two numbers to get the ratio would give you 1.2. The bigger the ratio, the more efficient the cell is. Therefore, the smaller cell with the ratio of 6 is more efficient. If you were to split up the larger cell into many smaller cells, the last picture on the right, the surface area to volume ratio would increase. So instead of having larger cells, it's in the best interest of the cell to have more cells. Cells, especially cells that need to move a lot of materials across the cell membrane, have evolved adaptations over time to accomplish this. One way that the cell can increase its surface area and therefore be more efficient at moving materials across the membrane is to modify their shape. Becoming skinny and elongated will increase the surface area to volume ratio. Also, some cells have long, thin projections coming off of them, such as microvilli in the small intestine, to absorb more nutrients from food, or root hairs to take in more water for the plant. So when you see a cell with projections or folds in the membrane, more often than not, these funny-looking structures are there to increase the surface area. Just keep that in mind. Remember, structure is related to function. While we will discuss structure and functions of prokaryotic cells later on in the course, when we look at how we can use bacteria in biotechnology, the majority of this chapter is dedicated to eukaryotic cells and its organelles. If you remember, at the end of the enzymes episode, I very quickly talked about how the cell is not just a big bag of enzymes, and the organelles make up different compartments so that there are spaces for each process and the enzymes involved in that process to do their work without interfering with anything else. Those membrane-bound organelles that are characteristic of eukaryotic cells is what divides the cell into compartments. Inside each compartment is a local environment that is specific to the process it's responsible for. So the cell can have multiple processes, each with their own enzymes and specialized environments going on at the same time. So this is where I'm going to stop for this first episode. In the next episode, we'll start going through the parts of the cell one by one and how they interact with each other to accomplish their functions. We're also going to compare and contrast animal cells, like this one here, with plant cells, like this one. As always, if you have any questions about this episode, make sure you remember to ask questions at the beginning of class. Otherwise, be a good human.